Good morning, good afternoon, and good evening, everybody. Welcome to a new Global IMMUNOTALK. Before we get started with today's talk by Bruno Silva Santos, just would like to remind you that next week we will have Adrian Liston, and we're here today with one of the organizers of the Global IMMUNOTALKs, Matteo Iannacone, who will introduce our speaker. Thank you, Matteo.
Thanks a lot, Carla, and it is my distinct pleasure to introduce Bruno Silva Santos as today's global immuno speaker. Bruno is a group leader and deputy director of the Instituto de Medicina Molecular, IMM, and professor and director of immuno-oncology at the Faculty of Medicine of the University of Lisbon in Portugal. Bruno is actually originally from Lisbon, and he did his PhD in immunology at the Cancer Research UK, the London Research Institute, and trained as a postdoc with Adrian Hayday at King's College in London. And that's where his interest in gamma-delta T cells originated. He then established his independent group at IMM in Lisbon in 2006. And has since received multiple grants and awards, in particular the starting and consolidator grants from the European Research Council. For the non-Europeans, these are the most prestigious grants in Europe, the size of which is often multiples of the NIH R01s. He has been elected as an EMBO member, the European Molecular Biology Organization, as well as honorary member of the European Academy of Tumor Immunology. And Bruno's research has been mostly dedicated to gamma-delta T cells, having dissected the molecular mechanisms of their differentiation, the way they recognize tumor cells. And he published his work in more than 100 scientific papers in the most important scientific journals. Besides his academic interest, Bruno was also able to develop a new technology for adoptive cell therapy named DOT cells. And he set up a startup company called LymphAct that has been recently acquired and is becoming a part of the Takeda pharmaceutical company portfolio. And a phase 1 clinical trial with the DOT cells in cancer patients is currently ongoing in the US. So Bruno, thank you very much for having accepted our invitation. We look forward to your talk. But before we start, as you know, we like to ask our speakers a question to get to know them a little better and to inspire the young generation of immunologists. And the question we would like to ask you is if you can describe a moment, a decisive moment in your career.
Thanks very much, Matteo. First of all, thank you very much to you, to the other organizers of this wonderful webinar series, and also Elena and Carlo, who started this off a couple of years ago. About the decisive moment, I must say, you mentioned Adrian, of course, and my focus on gamma-delta T cells, and my decision to join his lab has really obviously changed my life and my career, not only because of everything I learned, but also to get him as a wonderful mentor and friend that has inspired me throughout these years. So that choosing the right postdoc supervisor that can become a mentor is something that I find very important for everyone's career.
I cannot agree more. Thanks, Bruno. Okay, so without any further ado, you can share your screen and the floor is yours.
Thank you very much once again, Matteo. So hello to everyone. Of course, it's a huge pleasure, as I was saying, to join this wonderful webinar series. And I'll be telling you about the differentiation of gamma-delta T cells. And to start off, may I remind you that this dichotomy that all of us, I'm sure, know between B and T cells, namely alpha-beta T cells, that are by far the dominant T cell subtype in all jawed vertebrates, actually became a triad in the mid-80s when gamma-delta T cells were discovered. This was due to accidental cloning of the gamma and delta TCR genes. And one of the key players in this identification was precisely Adrian Hayday, who I just mentioned. And here you can see a picture of us, myself, Adrian, and a key collaborator throughout the years that I'll mention during this talk, Dan Pennington. And this picture was taken right at the end of my postdoctoral training with Adrian back in London. And so gamma-delta T cells became the key, let's say, in my scientific career. And you may know that gamma-delta T cells are quite unconventional, and this justifies the fact that they are non-redundant compared to alpha-beta, mate cells or NKT cells, all of these other T cell types. As you know, alpha-beta T cells depend on MHC presentation of peptidic antigens, CD1 presents antigens to NKT cells, MR1 to mate cells, and most gamma-delta T cells, the vast majority of gamma-delta T cells, does not rely on presentation by any of these presenting molecules. And actually, one of the major breakthroughs in the last decade has been the identification of a B7 B7-like family called butyrophilins that play important roles in gamma-delta T cell recognition, gamma-delta T cell activation, but I do not work specifically on the activation of these cells via butyrophilins, and so I will not mention this throughout my talk, but really invite you to visit and to get to know this fascinating family of proteins. So in my lab, what we do study is the early development of these cells in the thymus, and then because they migrate to many different tissues, you can see here in the mouse that actually several waves of gamma-delta T cells are generated throughout mouse ontogeny, and they populate very different tissues in the mouse. And actually, this picture from our most recent review on the topic is even more clear that you have an initial wave, or the mouse has an initial wave of gamma-delta T cells that populates the epidermis, and then you have a subsequent wave, and these waves, as you can see, are characterized by different V-gamma genes that are used in the rearrangement of the T cell receptor to compose that TCR. And so after a V-gamma 5 wave, there is a V-gamma 6, a V-gamma 4, a V-gamma 7, and later on a V-gamma 1 like wave. And what you can see is that these waves can actually, these cells can populate many different organs. And it's quite striking, the variety of organs where these cells can actually reside. And of course, if you think of barrier tissues like the skin and the gut or even the lung, the presence of these cells in the tissues can protect them against infections. And for instance, the roles of gamma-delta T cells against Staph aureus infection in the skin, or Listeria in the intestine, or Streptococcus or influenza in the lung are well established. These are conventional, let's say, immune functions of a T cell type. What is more unconventional, and it was once again a major development over the past decade, is the fact that these tissue, we can call them intrinsic T cells. We all know that tissue resident memory cells are now very well studied throughout the world, and gamma-delta T cells actually live in these tissues. They are tissue intrinsic, if we can call them that, and therefore they can contribute to the normal physiology of the tissue. So even in the steady state, what has been shown is that gamma-delta T cells can really participate in the normal physiology of skin, intestine, gingiva, the bone, the adipose tissue, or the central nervous system. And this actually was the major contribution of our lab to this new paradigm through the work led by Julie Ribaud, that really she showed, and her team showed, that gamma-delta T cells producing IL-17 in this case, and we'll go back to the cytokine, can really control the amount of brain-derived neurotrophic factor, BDNF, that is critical to ensure synaptic plasticity and learning in mice. And so this has been widely reviewed, and I would invite you to visit this literature if you're interested in this crosstalk between the immune system and the CNS, or this area of neuroimmunology. I should say, actually, that we just had Johnny Kipnis the previous week. Jonathan Kipnis' lab published a paper shortly after ours showing, again, that gamma-delta cells making IL-17 are very important for CNS function. So this is a new paradigm that has arisen recently. And then some unpublished work from our lab that you can check on bioRxiv actually shows that this importance of gamma-delta T cells in tissues can even be seen across generations. So I will just briefly summarize this study in one slide, but this study led by Pedro Papoto in the lab really showed that gamma-delta T cells can control, in this case, the microbiota that is transmitted at the maternal neonate interface. Essentially, what you can see here is different compositions of the microbiota of the babies, depending if they were derived from mothers either sufficient for Gammadelta T cells or deficient for Gammadelta T cells. And you can see very striking variations in several genuses and species of bacteria. That what is mostly interesting about these changes is that they associate with differences in the amount of short-chain fatty acids. And these are very important molecules to regulate immune responses. And you can see here that pups that were born from Gammadelta deficient mothers in white have less acetate, pentanoic acid, and other short-chain fatty acids. And this deficiency in short-chain fatty acids that tend to regulate immune responses and to keep them at bay actually means that the pups born from these mothers have exacerbated type 2 responses, both in the steady state, and this is the lung I should mention, both in the steady state and also upon parasitic infection. And so these dysregulated type 2 responses that come from this absence of the Gammadelta T cells in the mothers that generated these infants really can have implications in immunity and also in possibly in allergic reactions. This is a line of research that we'd like to explore further. And so I hope this introduction really tells you a bit how important Gammadelta T cells can be in several scenarios. And in our lab, a critical scenario for us is actually cancer and cancer surveillance. So we look at Gammadelta T cells within tumors, and we go, as Matteo alluded to in his introduction, all the way to developing immunotherapies based on Gammadelta T cells. And summarizing in this table a few studies that we published in the last few years, as you can see, like everything in immunology, nothing is good or bad, depends on the context. And so we can have Gammadelta T cells being highly protective or beneficial in several infection scenarios, cancer, and in neuroimmunology, as I alluded to, in cognitive functions. But if these responses are dysregulated or have a different quality of response, then the same cells, the Gammadelta T cells as a family, can behave as pathogenic. And so overactive Gammadelta T cell responses are at the very core of severe malaria and in particular cerebral malaria, but they can associate with tumor promotion and cancer progression. And in neuroimmunology, a dysregulated Gammadelta response can really promote the mouse model of MS, EAE, and recently we published that they can really foster the onset of Alzheimer's disease in a mouse model of AD. And what's interesting is when we try to look for the molecular mediators of all these functions of Gammadelta T cells, we invariably got one of two molecules as the mechanism. And these two molecules are very well known to most immunologists because they are very important cytokines, interferon gamma and IL-17. And this takes us back over a decade ago when Julie, she basically started a postdoc in our lab, actually identified two subsets of Gammadelta T cells from very early ontogeny. So even in the embryonic thymus, these Gammadelta T cells could be subdivided into two subsets, and we use the cell surface marker CD27 to discriminate between them. And as you can see, the CD27 negative cells were highly enriched in IL-17 producers, and they were really devoid of interferon gamma, whereas the CD27 positives had the opposite phenotype. So they contain interferon gamma producers and no IL-17. And so this functional dichotomy, we and our collaborators, once again, Dan Penton, Adrian Hayday, we coined it developmental pre-programming, because it means that in the thymus and even in the embryo, before the mouse has gone through any decent challenge and before the cells have migrated to the periphery, they have already decided which will be the key cytokine they will produce and engage in immune responses later on. And so this developmental pre-programming was actually shown to be a property not only of Gammadelta T cells, but by other groups of NK T cells and MAIT cells. And what these three T cell populations have in common is these innate-like functions. They are often called innate T cells or innate-like T cells, and they really respond extremely rapidly to challenge, possibly because they have been programmed since their ontogeny, since generation in the thymus, and so they are ready to go like an innate immune cell would be, without the need for activation, differentiation, clonal expansion of conventional lymphocytes. And so one of the issues that arose then was really how these potential progenitors of gamma delta T cells that were still immature would commit either to interferon gamma or IL-17 production in the thymus. And these early progenitors can be marked by expression of molecules such as CD24 or CD25. And what basically was very important to realize was that the T cell receptor itself, the signal that these cells that express a gamma delta T cell receptor would receive early on in the thymus, would dictate their developmental fate. And basically Miguel in the lab showed that the strength of signal would promote, strong signals promote the development of interferon gamma producing gamma delta T cells at the expense of the IL-17 producers. And actually, then Pennington's lab showed very clearly that strong TCR signals are very detrimental to IL-17 producers during thymic development. And so it's the interferon gamma producing cells that benefit from strong agonist selection in the thymus. Naturally, the TCR is at the cell surface and it only provides these kind of developmental consequences because it creates a series of downstream effects all the way to the nucleus. And so we were involved also in dissecting what in the nucleus could regulate the differentiation of these two subsets of gamma delta T cells. And to cut a long story short, we and other labs really have dissected a lot of transcriptional mechanisms that account for the development of IL-17 or interferon gamma producing gamma delta T cells. And although they use very often these master transcription factors like Rho gamma T for IL-17 producers or TBET for interferon gamma producers, it's worth noting that gamma delta T cells and this pre-programming in the thymus also involves transcription factors that are lineage specific and that ensure that these master transcription factors that regulate the cytokine expression can be prematurely expressed and stably regulate the expression of the cytokine genes. This is the case for SOX13, for instance, which is really a gamma delta biased gene that together with CMAF guarantees that Rho gamma T is there activating the IL-17A transcription in these early gamma delta T cells. But so we and others have dissected a lot of transcriptional mechanisms. And so the first story that I would like to discuss with you today briefly is the importance of post-transcriptional regulation to the differentiation of these two subsets. And basically, you all know that when mRNAs leave the nucleus, they can be regulated in particular by microRNAs. This is one of the major mechanisms of post-transcriptional regulation, and microRNAs essentially they are these small non-coding RNAs that can bind to target DNAs based on sequence homology, and by doing so they will repress the translation of these mRNAs or even lead them to degradation, and therefore decrease the protein expression in the cells. And so microRNAs can be extremely important to fine-tune gene expression so that you don't rely just on transcription and on transcriptional complexes to dictate how much transcript you have, but you can then fine-tune with these levels of these microRNAs that enter between mRNA and protein to dictate levels of expression. And this project started with a very obvious phenotype of mice that we analyzed, mice that lacked microRNAs and looking at their gamma delta T cells. Basically, these mice lack microRNAs specifically on T cells because the deletion of this enzyme called dicer, which is essential to generate mature microRNAs, this dicer has been ablated just in cells that express LCK. And LCK, you may know, is the proximal signaling molecule for the T cell receptor, and therefore it's in T cells that dicer will be deleted. And when you look at gamma delta T cells, what was very striking, and looking here more specifically at the gamma delta cells that are CD27 negative and therefore in control mice will be committed to IL-17. We here used other markers to really have a very enriched IL-17 producing population. What you can see is that in the absence of microRNAs, there is a complete dysregulation of cytokine production, because now these cells, instead of being pure IL-17 producers, they acquire interferon gamma production, and they actually decrease overall their amount and their IL-17 production. So you have many more cells that are double negative, as you can see, and you also gain double producers IL-17 interferon gamma. So this means that microRNAs can really regulate cytokine expression in gamma delta T cells. But in the mouse, there are 2,000 microRNAs. So which microRNAs could be involved in this kind of phenotype became the question for us. And we actually identified the first microRNA that is important functionally to regulate this cytokine expression. This was published a few years back, so I'll just show you this little summary where we identified one microRNA called 146A, which, as you can see, kind of mimics the phenotype that I showed you for the overall microRNA deletion, which is a dysregulation of cytokine expression in the cells that normally would just make IL-17, but they're now also making a proportion of them, also making interferon gamma. So what this paper showed is that these cells normally have an intrinsic break, which is 146A, this microRNA, that prevents them to go down the interferon gamma pathway. But if you ablate this microRNA, then you allow cells to actually acquire interferon gamma as well. Okay, we have one out of 2,000. So the next question is, can we have a bigger view and a more decisive view on how many microRNAs can be important for gamma delta T cells, in which directions and in which contexts? And to address this question, we decided at that stage to go from surface markers that were already extremely useful to have a more specific strategy to focus on cytokine producing cells. And so for that, we generated a double reporter mouse where IL-17 producing cells express GFP as well, and interferon gamma producing cells express YFP. And so these are alternatives. As you can see, you have an iris sequence there. So the cell will have an intact IL-17 or interferon gamma protein, but together in the same cell, a fluorescent protein. And so you can use this fluorescence, obviously, to sort the cells. And you can see this system is extremely clean, because when you look at the gene expression level, it's only the GFP positive cells that have any transcripts for IL-17A. The YFP or the double negative cells are completely devoid, and the reverse for YFP positive cells, only they express interferon gamma. And this is not just the signature genes. Just to give you an example, all the other genes that associate with IL-17A as CCR6, IL-17F, IL-22, etc., they are all very much expressed on those cells, the green cells, and not in other gamma delta T cell subsets. So using this very clean strategy now, we could inquire the full microRNA, let's call them the microRNA transcriptome of these different subsets. And the conclusion was, overall, that out of 2,000 microRNAs, around 100 of them are differentially expressed between the two gamma delta T cell types. Around half of them going towards the IL-17 and half of them going towards the interferon gamma subset. But of course, we have 100. How can we screen them and get the most meaningful microRNAs that could even act as therapeutic tools to regulate cytokine expression, not only in preclinical models, but more importantly in humans, of course, as a proof of concept for humans. And so to do that, we basically modulated in systems, in vitro systems, the expression of these microRNAs by retrovirally infecting the gamma delta T cells with constructs having these microRNAs of interest, or providing mimics for these microRNAs, so different strategies to overexpress the microRNA, and we inquired and checked the impact on interferon gamma or IL-17. And especially interferon gamma, you can see here, was clearly modulated by this overexpression strategy. You can see in these FACS plots here, this histogram, that we have, for instance, 3:2, this microRNA in pink, that increases the expression of interferon gamma, which is what we are analyzing on the x-axis compared to controls in gray. In gray, you have the empty virus. And then you have blue and green for 2 microRNAs that clearly reduce the normal expression of interferon gamma in the CD27 positive gamma delta T cells. So individual microRNAs can modulate the expression of interferon gamma even in opposite ways. And to give you a bit more granularity about some of these candidates, 1-8, for instance, that really downregulates the interferon gamma, and 3-2 that upregulates, I can show you this other experiment where we really tracked both the percentage of cells or the MFI in independent experiments, all of them agreeing with an increase with 3-2 of interferon gamma and a decrease with 1-8, which is very obvious as well at the MFI and in this histogram. And this is not only for those cells that naturally make interferon gamma, but again, we can even interfere with the production of interferon gamma and its normal absence in the IL-17 producers, because this 3-2, the microRNA that increases interferon gamma, does so even in IL-17 producers, forcing them, as you can see here, some of them to acquire interferon gamma expression. And what's very interesting about this kind of microRNAs, once again, going back to this paradigm of developmentally programming these gamma delta subsets, is that these microRNAs start being expressed in early thymic development. So here you have CD24, this marker for early gamma delta cell progenitors, and you have embryonic thymus, newborn thymus, and then adult lymph node. And you can see that it is the thymus, the pink and the blue, and actually the earlier the thymus, the highest the expression of this particular microRNA, 1-8, that really express high levels of the microRNA that then can be downregulated along effector differentiation, especially because in this case, this is a microRNA that inhibits interferon gamma production, and therefore you can downregulate it to acquire interferon gamma functionally. And this same pattern applies to a different microRNA, in this case 1818, that also is highly expressed in the thymus. And interestingly for this microRNA, because we could access mice deficient for these microRNA in particular, it was very interesting to see that compared to litamate controls, the phenotype of these mouse that lacks this one microRNA is that gamma delta T cells have less IL-17 and they have more interferon gamma. This, as you may remember, was the general picture I showed you for the DICER deficiency, the absence of microRNAs, and we find one microRNA out of 2000 again that can really mimic that phenotype in partially. Obviously, this is one out of several that can potentially have this effect, but it's very clear the effect that this one microRNA can have. And this one, it's particularly interesting, 181A, because we think it can really be involved in this major mechanism of differentiation that I mentioned earlier on, which is the strength of signal that comes from the T cell receptor dictating the fate of the gamma delta T cells. And why do I say so? Because in alpha beta T cells, this same microRNA was shown to modulate T cell sensitivity and selection. And so this now becomes critical for us to put in the context of gamma delta T cells, since they need to go through strong TCR signaling to become interferon gamma producers. And so the capacity of this one microRNA to target these very important phosphatases, so what you have here in red are names of 4 different phosphatases that are direct targets of microRNA 181A. They have the homology sequencing, the seed sequence, that complementarity that will allow this microRNA to bind and to regulate the expression of these phosphatases. And what are these phosphatases? Well, they are key regulators of LCK and ZAP70, these key kinases of proximal TCR signaling, of ERK, that, as you know, is a key MAP kinase enzyme, and therefore downstream of this early TCR signaling, and also PTEN, which is a regulator of PI3 kinase signaling. And so these very important signaling pathways, all of which are required for strong TCR signaling, can really be manipulated and fine-tuned by this one microRNA. So we think this is at the core of the phenotypes we've analyzed. But of course, we want to have a much bigger picture, since we now have the 100 microRNAs differentially expressed between the 2 subsets. And naturally, we also did mRNA sequencing to know how the different genes that can be involved in, namely transcriptional regulation of this differentiation, change between the 2 subsets. And so we can try, and this is what has been done in this slide, in silico, to match and to create microRNA, mRNA networks. Which microRNAs that are differentially expressed may be targeting the mRNAs that are differentially expressed, and therefore try to construct these networks that we hope will really shed a lot of novel insight about the fine-tuning and regulation of differentiation of gamma delta T cells. So this, as I mentioned until now, has been in mice, but let me show you that this is relevant in humans. So I'll just very briefly allude to a paper that just came out, a study led by Julie Ribaud. And Julie, basically, with Gisela, a PhD student, what they've shown now for human gamma delta T cells, they differentiate them also starting with naive cells. So we can do this because we can get thymic biopsies from children that undergo cardiac surgery for cardiac reasons. And therefore, the thymus that needs to be removed to access the heart can be used, and it's donated for, in this case, to do this kind of experiments. And what you can do is basically to differentiate the cells along the type 1 pathway. And it's very clear, as you can see in these correlations, how the more microRNA 181A you have, this is in the x-axis, as you increase the expression of the microRNA, you decrease the interferon gamma production, and also NKG2D, which is a master gene for cytotoxicity of these cells. And so this type 1 cytotoxic profile of human gamma delta T cells that is critical for their anti-tumoral functions is, as you can see, downregulated by this microRNA. And this we think is very interesting because when you look for what could be driving this high expression of the microRNA, the cytokine that induces really high expression of this microRNA is actually TGF-beta. And you know, TGF-beta is highly immunosuppressive. It can accumulate in tumor microenvironments. And therefore, we think this kind of knowledge is really pointing towards an immunosuppressive mechanism in tumors in particular, where TGF-beta can upregulate the expression of a microRNA that will shut down the effector program of human gamma delta T cells. And because this just came out a couple of months ago, I will not tell you more about this and invite you to read the paper if you're interested in this kind of topic. But now I'll shift gears to finish my talk. The second half of my talk will not be on microRNAs, but back to mRNAs that I guess everyone is more familiar with, obviously. And we ask, to begin with, the same question. Out of the, in this case, 21,000 genes that are present in the genome, how many of them are differentially expressed between the two subsets of gamma delta T cells isolated from this double reporter mouse? And the answer was quite striking because almost 8,000 genes, although these are both gamma delta cells, very similar until a certain point where they diverge functionally, 8,000 genes are differentially expressed above twofold between these different subsets. And so what was very interesting, we have lots of genes to really look at, as you can imagine, but from a helicopter view and trying to do pathway analysis, when we did KEGG analysis of different pathways, what was very obvious was this hint of metabolism being very different between the two subsets. As you can see, metabolism, be it of amino acids, of carbohydrates and lipids, all of these metabolic pathways seem to be differentially enriched in the two subsets, justifying these data from the RNA-seq experiments. And so Noëlla Lopes, that was a postdoc in the lab, now working with Eric Vivier back in Marseille, Noëlla decided to take those data and to really validate it at individual genes focusing on metabolism. And as you can see, looking first at glycolysis-related genes, it was very clear that the gamma delta cells making interferon gamma, which are here in red, express higher levels of these key enzymes of the glycolytic pathway. So these are the genes we are looking at, and as you can see, always higher expression in the interferon gamma compared to the blue cells, the IL-17 producers. And besides the enzymes themselves, what was clear was that this transcription factor, and very important oncogene, MYC, was really very highly expressed on the interferon gamma producing gamma delta cells. And why was this so interesting is because the genes that encode glycolytic enzymes are direct targets of this transcription factor. So MYC regulates glycolytic enzyme expression. And so it it was extremely provocative that we had such a clear transcriptional difference between MYC expression in these two subsets. So it was clear that glycolysis was, let's say, enriched, at least at the gene expression level for now, in the interferon gamma producers. But what about the IL-17 producers? So it was there very obvious as well that we had the opposite phenotype when we looked at mitochondrial genes. So now looking at components of the complex 1, complex 2, complex 4 of the mitochondrial electrotransport chain, if you look at these genes, you have exactly the opposite, higher expression in the IL-17 producers in blue compared to interferon gammas. And again, quite interesting as a master regulator, this nuclear respiratory factor 1 and RF1, which is a key transcription regulator of the mitochondrial biogenesis and these kind of genes, was differentially expressed very strikingly towards IL-17 producers. So it seemed really at the gene expression level, we had a beautiful dichotomy between glycolysis and OXPHOS. But of course, we needed to validate this functionally. And the problem there, as you may know, gamma delta cells are not the most abundant cells, and the typical techniques used for metabolic studies, like seahorse flux analysis, required a lot of cells, up to 1 million cells very often. And so we really needed to use something different to analyze the numbers of cells we would get from lymph nodes, from tumors, and from the thymus itself to analyze these energetic profiles. And this was critical, a collaboration with Rafael Arguello in Marseille. So Rafael developed a new, and that I really think is something you should take from my talk today, because it can be extremely helpful for all your metabolic studies that you may wish to do at a certain stage in your project, because this really is a very easy assay and extremely informative. Basically, it uses a property of puromycin, which can intercalate in peptides as they are being translated. So very similar to BRDU that can intercalate with DNA and does allow us to look at intercalation of DNA and replication and cell division and proliferation. In this case, puromycin can take the place of a tRNA that is bringing amino acids into the nascent polypeptide. And this intercalation of puromycin makes all nascent peptides visible to an anti-puromycin antibody. So you use an anti-puromycin fluorescent antibody, and the MFI that you get for cells will tell you essentially how much protein translation is taking place. And because protein translation is by far the most energy demanding process in the cell, this will be a great proxy for your energy requirements and utilization in the cell. Naturally, to know which pathways are responsible for that energy, what you need to do is to use inhibitors, typical metabolic inhibitors, for glycolysis, for mitochondria, like oligomycin that really uncouples mitochondria, or combinations of these inhibitors, and you see the impact of this inhibition on your puromycin MFI. And then you just use mathematics to really calculate the dependence, the impact on that MFI. There's a series of formulas that you can really use to calculate how much your cells were dependent on mitochondria, or on glycolysis, on fatty acid oxidation, and so forth. And what you can see here was the validation of this gene expression characteristics I showed you at the functional level. So if we focus first on the IL-17 producers, which in this case are the solid bars, when you look at the mitochondrial dependence, you have very high mitochondrial dependence for the IL-17 producers, the solid bar. And when you look at the interferon gamma producers, essentially there's no mitochondrial dependence. So unlike the gene expression data, it seems the interferon gamma producing cells are not using mitochondria. By contrast, they increase their glycolytic capacity. So compared with the IL-17 producers, the solid bar, the stripy bar of the interferon gammas is much higher. So these are gamma delta cells from spleen and lymph nodes, and this validates the gene expression profile in the steady state. Naturally, and because these subsets, as I alluded to, can be extremely important in physiological responses, namely to cancer, we asked how these metabolic profiles would behave within the tumor microenvironment. And for that, Noella used two tumor models, a breast cancer a cell line, E0771, and a colorectal cancer cell line, MC38. And as you can see in both models, really within tumors, the tumor-infiltrating gamma-delta T cells, the two subsets, behave exactly like they did in the spleen and the lymph node, with a mitochondrial dependence for the IL-17 producers and a high glycolytic activity for the interferon gammas. And what was most surprising, that goes back to the title of my talk, is that this was already seen in the thymus and even in the embryo. So once again, the thymus where the cells are born, these cells are already diverging, in this case at the metabolic level, and getting an energetic profile that they will maintain in secondary lymphoid organs, in tissues, in tumor tissues, and upon infection, if that is the challenge. So this is really, these are hardwired metabolic programs that these two cell types use. And really to understand more how relevant for thymic development these profiles could be, our collaborator that I alluded earlier on, Dan Penton in London, who's even more fond of T-cell development than we are, and has great expertise using fetal thymic organ cultures, which are the best in vitro system to mimic T-cell development. Basically, you put fetal lobes in culture and you can manipulate those fetal lobes at will and see the output in terms of T-cell development. What Dan was able to show, his lab, was really that this mitochondrial divergence between cells can really dictate their fate. So what they've done was to use this TMRE, which is a dye that labels active mitochondria, to really subset the early gamma delta cell progenitors that express CD24 before they become interferon gammas or IL-17, before they have the cytokine, and really show that if they are low for mitochondrial activity, they will become, in this developmental assay, interferon gamma producing cells, which are here marked by two cell surface markers that correlate with cytokine expression that we also validated separately. So basically, low mitochondria, interferon gamma. If they take these progenitors with high mitochondrial activity, you get exclusively almost the IL-17 producers. And you can see this beautiful dichotomy: low mitochondrial activity, interferon gamma; high mitochondrial activity, IL-17. And these associate at the single cell RNA-seq level also with the expression of, once again, OXPHOS-related genes versus glycolysis-related genes. So the next question was really, what was happening? Why are these two subsets diverging metabolically early on in the thymus? So we realized that those progenitors earlier on, the earliest progenitors actually had high mitochondrial activity. They were like the IL-17 producers, which you can see here stain very well for TMRE, which is this dye for active mitochondria. Basically, these dyes are sensing the mitochondrial membrane potential. So when you have this staining, it means that there is a membrane potential, the mitochondria are active. By contrast, in the interferon gamma producing gamma delta cells, as you can see, there's no activity in these mitochondria. And so the progenitors, the early progenitors, had active mitochondria. So this meant that it was an active process of shutting down the mitochondria that was occurring in the interferon gamma pathway. And to inquire what could be driving this shutting down, we postulated at that stage, trying to link with our model of strong TCR signals promoting interferon gamma, we questioned if by giving these early progenitors strong TCR signaling with an agonist antibody for the TCR, if we could then really engage and shut down the mitochondria, if we could lead to a downregulation of this TMRE staining. And as you can see, that's exactly what we got in a dose-dependent manner. There was a pool of gamma delta progenitors that would downregulate TMRE, shutting down the mitochondria at the same time as they would acquire interferon gamma, which was the final readout of this assay. So really, basically cutting a long story short, this paper was published last year. What we showed in this paper is that upon strong TCR signaling, you get a metabolic shift early in the thymus that makes gamma delta progenitors, some of them that get these strong signals, to divert away from IL-17 production and become interferon gamma producers. What this strong TCR signaling does, that is critical to engage in high glycolysis as an alternative, the so-called aerobic glycolysis as an alternative alternative to OXPHOS is that these signals induce MYC transcription. So I showed you these beautiful RNA levels that are very black and white between the IL-17 producers and the interferon gamma. And here you can see the validation at the protein level using a MYC reporter mouse, that really you only have MYC in the gamma delta cells that express interferon gamma. So it's this wiring between strong TCR signaling, MYC expression, high glycolysis, and shutting down mitochondria that make cells have a very different metabolic programming throughout their life. So this happens in the thymus, we see it in the periphery and in tumors. So to finalize, the final question is this relevant? Namely, is this relevant for the responses that these two different types play in the tumor microenvironment? So I told you earlier on, and I should stress it now, that the two subsets are dramatically different in their impact on tumor growth. Because, as you can imagine, interferon gamma is a strongly anti-tumoral cytokine. So this subset of gamma delta cells is really anti-tumoral, whereas the gamma delta cells making IL-17 promote tumor growth. And we and others have shown this in many different models. So the final thing we wanted to ask here was, can we boost the anti-tumoral cells using this knowledge that we just got, that they are very dependent on glycolysis? And so what we've done first in vitro was to provide a glucose boost to these cells. We sorted the gamma delta cells making interferon gamma, these 27 positive. And as you can see, compared with control medium with low glucose, if we inhibit glycolysis, we really reduce interferon gamma from 37 to 14. But if we provide high glucose to this medium, we now have 65% of interferon gamma producers. So really glucose is a major booster of the interferon gamma response. And it's not just gamma production, because even the killing capacity of these cells, which is critical to handle tumors, of course, is also boosted by high glucose, which is what I'm showing here, upon co-incubation with that breast cancer cell line I mentioned earlier on. There's always much more killing of the tumor cells with the gamma delta cells exposed, in this case, just during 3 days to high glucose versus low glucose. So to translate this in vivo, what we've done was to use a protocol that Erica Pierce previously used in one of her papers, which is an even shorter glucose boost, just for 5 hours, to the cells that you're going to adoptively transfer into mice. So we have mice that get breast cancer. The breast cancer develops, and once the breast cancer is there and palpable, we can now adoptively transfer gamma delta that have received high or low amounts of glucose, or control cells. And as you can see, what you get is really a dramatic impact of the gamma delta cells that were incubated with high glucose, that can really control tumor growth, unlike the cells that got low glucose that provide very minimal impact in this tumor model. So this boosting, we think, can be dramatic in the context of adoptive cell therapy, of course. We are not suggesting you should provide glucose because the tumor itself will use glucose for its proliferation. And actually, it's known that the major tumor immunoevasion mechanism is tumor cells starve T cells from glucose and prevent them from being anti-tumoral cells. So in this case, we provide the glucose in vitro before the adoptive cell therapy. And this is what we would suggest, and I'll finish on that on a translational note. Let me just tell you that it is very clear from our research in collaboration with Dan that the anti-tumoral cells really thrive on glucose to promote aerobic glycolysis. And at that stage, it was quite unclear to us how the IL-17 producers would really fuel their mitochondrial activity. And it was very nice because Lydia Lynch, that will present at this webinar series later in the year, she was actually studying 17 cells were actually fueled. And she came with this very beautiful dichotomy that the IL-17 producers really uptake lipids very avidly, in stark contrast with the interferon gamma producers. So you have very high lipid uptake, and cholesterol uptake by these gamma delta cells making IL-17 drives their proliferation and their pro-tumoral functions in vivo. So it's this dichotomy that we showed in this paper that joined the work of our labs. And to finish off with a slide of translational implications, let me address the point that Matteo referred to. So we generated a few years back a protocol that is being used in a clinical trial run by Gammadelta Therapeutics, the British company that will soon be part of Takeda pharmaceutical company. And Gammadelta acquired our Portuguese pharmaceutical startup, LinkFact, where we developed this adoptive cell therapy protocol for human Gammadelta T cells. So with this protocol that is now being used in AML patients, we really can, as you can see in these plots, really expand a lot the Gammadelta T cells and endow them with very strong anti-tumor activity, high interferon gamma, high NK cell like activity against tumors. So we are now studying the prospects of intervening during the 2 weeks. Now it's a 2-week protocol. The 2 weeks where we are really manipulating these human Gammadelta T cells to consider metabolic intervention as a way to maximize the outcome of these cells once infused into the patients. And with this, I will finish my talk and really acknowledge the outstanding work that my lab has done, really an outstanding gang of people. Teamwork makes the dream work, and that is really our motto. And the team is not only our lab, but extended to these very important collaborators that I mentioned throughout the talk, then Penton, Lydia Lynch, Raf Arguello, Judy Allen in the study by Pedro that we are finishing off, and collaborators back at IMM, our funders. And really thank you, all of you. It was a pleasure to give you this talk.
Wow, thank you very much, Bruno. Fantastic talk, and what a beautiful journey on the immunobiology of Gammadelta T cells from basic science all the way to the clinic. So thank you. So let me just remind everyone, let me share the screen, how to ask questions to Bruno. You can actually, the questions, as in the tradition of the Global IMMUNOTALKs, will be asked via Twitter. So you can search for the account Global IMMUNOTALKs. You can find the tweet that says, ask questions for Dr. Bruno Silva Santos here. And you should reply to that tweet with your question. And you should mention also the Global Immuno hashtag. And Bruno will reply with his own Twitter account. So thanks again, Bruno, and thanks everyone for listening.
Thank you. Bye.